Hello everyone, and welcome to our webinar called Oncolytic Virotherapy for Pediatric Solid Tumors. It's presented by Solving Kids Cancer. My name is Scott Kennedy. I'm the co-founder of Solving Kids Cancer. Here at SKC, our mission is to improve survival for children with neuroblastoma, brain tumors, and solid tumors by supporting the launch of novel clinical trials that expands the menu of options for these kids who really have very few options. And our goal today is really to open a discussion about a very exciting and potentially very promising area in pediatric cancer treatments called Oncolytic Virotherapy, which utilizes oncolytic viruses. This year in 2011, there's going to be 5 different oncolytic viruses in clinical studies throughout the U.S. for children with cancer. The new agents are really at the cutting edge of the treatment paradigm for children today. And Solving Kids Cancer feels it's important to have families and researchers in the same virtual room, so to speak, to really open up a discussion about this. Full disclosure, Solving Kids Cancer has invested our research dollars into supporting 3 of the 5 agents we're going to talk about today. What's most important is that there are as many new options in an exciting new category like oncolytic viruses as possible, and each of the researchers will talk about the various differences and some of the various challenges that have come about and why it's taken so long to get a therapeutic like this into the clinic. I'd like to thank all the parents from all over, as Donna said, all over the world who made the time to participate today, and all the many folks from the cancer research and medical oncology community who are also joining us today. I'm thinking that about half the attendees, I think we have over 120, are parents, and maybe half of them are other researchers from all over the world. So it's very exciting to have both important audiences in the same room. To all the researchers in the audience, a little side note, we'd like to invite you to submit a clinical trial proposal to Solving Kids Cancer for review and potential support. You can do this by visiting our website and reading our guidelines, and we accept proposals throughout the year. On to the meeting. We're really honored to have 4 pioneering pediatric oncologists who are the principal investigators of these 5 different oncolytic viruses. And they're going to present overviews of these new therapeutics and take some time to answer some of your questions. So without further ado, I'd like to take care of some of the housekeeping duties and provide a basic overview of how this webinar is going to work. Our primary goal of the meeting is to run at about 1 hour and 15 minutes. It's sort of an aggressive agenda with 4 different presenters presenting 5 different areas. We're going to reserve the last 30 minutes for questions and answers. The audio portion is muted for all the participants, which means that you can hear the presenters, but they can't hear you, and we do this to eliminate potential background noises. The call and the presentation itself is recorded, and we're doing this to provide a replay archive of the entire webinar for everyone. And what we'll do is we'll provide a follow-up email within the next 2 days or 1 day to all of you, which will have the link to the archive. And for those that weren't able to attend but registered, we'll follow up with them and they'll be able to view a replay of this webinar. And our goal is to really make this an interactive meeting, so we really welcome your questions and comments throughout the meeting. And the way we're going to handle that is to have you type your questions in the Chat with Presenters section, which you see at the bottom left of the page. And you can do that throughout any portion of the meeting and click the Send button right to the right. And just make sure not to use the raise hand function, which is on the top. That's not how we're going to be answering questions throughout the meeting. We'll stick to just using the Chat with the Presenter section. We'll collect these questions throughout the meeting, and along with a few other standard questions we've assembled for the researchers, we'll cover these questions. And those that we don't get to cover, we're going to follow up with the researchers and provide them in a Q&A format in an email that we'll follow up with you, the same email that will have the archive link in. Lastly, if there's any technical problems, please go back to your confirmation email we've sent 2 or 3 confirmation emails. So either any of those, the most recent one was sent yesterday, will have a phone number and email address for technical support and potentially an online chat with ReadyTalk to be able to answer your technical support questions to get back into the call. So far, we've been a very robust system here. It's very ambitious to have so many participants, but we love the technology that allows us to be able to have 4 researchers in the same room talking to over 120 people from all over the world. So it's very exciting. Thank you again, everybody. And without further ado, we're going to start the presentation with Dr. Timothy Kripe. Tim is going to be doing a general overview of oncolytic viruses, and then he'll go and describe 2 clinical trials that he's the primary investigator of later on in the presentation. A little background about Tim. He's a pediatric oncologist and professor of pediatrics at Cincinnati Children's Hospital, which is affiliated with the University of Cincinnati. He directs a research lab that studies novel biotherapeutics with an emphasis on oncolytic viruses. He's been truly a pioneer in oncolytic viruses for over a decade, and the first that I know of to bring it into the clinic for pediatrics. Tim co-authored a recent overview on oncolytic viruses that was published in 2010, and he's also in the midst of writing a book on the subject of oncolytic viruses. So Tim, I'd like to start with you.
Thank you, Scott, and thank you, Donna and John and everyone else at Solving Kids Cancer for sponsoring and hosting this webinar. It's very exciting for me. This is my passion, bringing new viral therapies to children with cancer, so thanks on behalf of all kids with cancer. This is a daunting topic. There's a lot to cover, and in some ways this will be just an introductory webinar and somewhat superficial. And I know I can see the list of people that have signed in. I know there's some pretty robust scientists, including, in addition to parents, some whose names I recognize, and hopefully this won't be too basic, but I think it's important to start with a foundation. Certainly some of what I'm going to say is a bit simplified in order to get the point across, but we can always discuss the nuances later. So without further ado, let me advance to my first slide. And really the story in some ways goes back to George Dock, who was a professor of medicine in Ann Arbor and then dean of the school at Washington University School of Medicine. And I start with a little history because I think it's always important to understand where we've come from in order to inform better where we're going. And what Dr. Dock's contribution was is listed on the bottom of that slide, a publication in 1904, The Influence of Complicating Diseases Upon Leukemia, where he presented a patient at a conference and then in this publication. And the patient presented to him in 1896 with obvious leukemia, a white count of 367,000, a bone marrow that was packed with blast cells. And he followed her because at that time there's really no effective therapies, and noticed that she got sick with what he diagnosed with as influenza. Of course, the fact that influenza was caused by a virus wasn't known then. In fact, viruses were just being uncovered, the tobacco mosaic virus being the first virus in a plant. And so it wasn't for another 30 years or so before influenza was even identified as being a virus. But nevertheless, he was able to make that clinical diagnosis and noted on subsequent visits that she had apparently gone into a clinical remission from her leukemia. And unfortunately, this was only a transient remission, but it struck him nonetheless as fairly dramatic. And he cited looked through the medical literature and realized there were a number of other cases dating back into the mid-1800s of similar cases of patients with cancer who, of course, at that time had no real treatment options, who subsequently suffered from either bacterial or viral infections and had remissions. Again, most of those were temporary, but nevertheless, this prompted him in an address at the Academy of Medicine in Cleveland, Ohio in 1903 to postulate that it might seem that the process could be imitated and a symptomatic, if not a causal treatment be discovered. So in many ways, I attribute the birth, or I ascribe the foundation, the beginnings of this field to Dr. Dock from that that insight. So the question was, could we take advantage or exploit this, what appeared to be an experiment of nature? And this was quite an exciting concept at the time. So the point here is it's not a new idea, and people have been trying to harness some of the immune modulating effects and the cytolytic or cell killing effects of infectious diseases to fight cancer for a long time. And this took hold in even as far back as the 1920s when animal tumors were being studied. This is a figure from a paper that was published in 1922 by Constantine Levadetti and Stéphane Nicolau from the Pasteur Institute in Paris, where they were able to show that when they infected herpes virus lysates or collections of cultures into tumors, and these were rabbit epitheliomas, skin cancers, they were able to have that virus grow in the tumor, and they were able to subsequently show that the virus that grew in the tumor was able to induce other virus-like diseases in other animals. So there's a little picture of a brain. Let's see if I can get the pointer there, and showing that the culture could induce encephalitis as well as keratitis, another known herpes virus infection of the eye. There's a drawing of a cornea there. And he also noted in a follow-up paper that was published showing that the tumors that were infected by viruses grew more slowly or in some cases regressed. And so that was, I think, the first that I know of an animal model of a cancer that was infected and showed some anti-tumor effect. And why would viruses like to grow in cancer cells? And so this slide is my depiction of sort of a very basic watered-down concept of why this might be the case. So cells have, if you think of this box as a cell, cells have a natural resistance to virus infections, but they have a couple of properties that make them want to resist virus infections. Or normal cells that are differentiated, that aren't dividing, have a low metabolism in general, and they also have innate virus defense mechanisms, most notably the interferon pathway. So interferon is a small protein that was described originally as a protein important for virus for defending against a virus infection. And it also has a lot of normal functions in the cell, most of which are controlling cell growth. So how does a cell become susceptible to virus infection? Most viruses that are capable of infecting normal cells encode their own genes to rev up the metabolism of tumor cells, so to create the cell factory machinery for reproducing more of itself, making more DNA or RNA, and making more proteins to produce more virus. So the virus genes are designed to do that. And virus genes also are designed in many cases to interfere with the interferon defense mechanisms of the cell. So they produce genes that counteract at various trigger points the cellular defense mechanism. So by doing those two things, viruses can infect and grow well in many normal cells. And it turns out that those two characteristics of a cell are also two characteristics of cancer cells. So cancer cells, of course, are growing. They need to produce a lot of nucleic acids by copying their own nucleic acids, and need to produce a lot of protein to make new cells and to grow. And they also, turns out, need to inactivate somewhere along the line the interferon response, because much of the proteins involved in the interferon response are there to regulate cell growth. So that unregulated cell growth that is a characteristic of cancer cell, both the increased metabolism and the defective interferon response, leads them susceptible to virus infection. So in these ways, we can thus either find viruses that grow well in tumors cells, or engineer them by either deleting their ability to fight off interferon, and therefore they'll be more restricted in their growth of normal cells, but still be able to grow in cancer cells. So this idea was took root in many ways, and in fact in the 40s there was some investigators, mostly at Memorial Sloan Kettering in New York City, and also around the world actually, that decided to start trying injecting patients with viruses. Now, at the time, in fact, there was very little oversight, regulation, very little quality control. Some of the investigators were just taking crude cultures of viruses and injecting them into patients, and even taking some that grew out of one patient's tumor and putting them into another patient's tumor. So this really was somewhat of a dark time in clinical trial history. Many of the events that unfolded resulted later in a lot of the regulations we have today for good reason. Things were although I think they probably had the best intentions in mind, without appropriate checks and balances and quality control, the integrity of the scientific data is in question, and there's also undue risk that happened. But they did see some tumor responses, but the viruses were not attenuated in any way, and patients often suffered from infections from those viruses. And so combining that with the fact that the trials were not regulated and were not well planned ahead of time, and the fact that chemotherapy was coming into existence and showing a lot of efficacy, the interest in this area waned dramatically until the modern era, when we were able to attenuate or engineer viruses to be more safe. And there's been quite a resurgence over the last 10 to 15 years in this field. And so I kind of listed on this slide 4 recent key developments. That is the ability to engineer viruses or to find mutants of viruses that are somewhat crippled in their ability to cause infections, but in the tumor cell that's already susceptible or that has interferon defects. These gene-deleted viruses or mutant viruses are unable to thwart those the interferon defenses in the normal cells. Again, they don't need to do that in a cancer cell, and so there's one way that these viruses have selectivity to grow in and kill tumor cells. We've also been able to discover a lot of non-pathogenic viruses, that is, viruses that infect people but don't cause disease. So they're too weak to cause disease, and so they're already, by definition, essentially safe for use in people. And in many cases, it's because they are susceptible to those interferon defenses, and they can't really mount a robust virus infection and cause a disease. But in the tumor cell that lacks those defenses by various reasons, such as RAS activation, other pathways, they're able to infect and kill the cancer cells that lack those interferon defenses. And then similarly, people have been studying animal viruses that don't infect people, and again, therefore, by definition, don't cause disease and may be safe to use in people. And so and it's been discovered that in many cases, these viruses are don't cause disease because of the interferon pathway, but they can infect cancer cells in people. And then finally, genetic engineering has allowed us to manipulate the DNA sequence or the RNA coding sequence of viruses to create our own mutants, to delete or add genes. And deleting genes, again, making the attenuated mutants, or adding genes to enhance effects. So one of the viruses we're going to talk about, the JX594, has a gene that stimulates an immune response with the idea that one can sensitize the immune system to tumor antigens. So these have really resulted in, I would say, almost an explosion of interest in this area. Viral therapy has gained public attention as well. There's a novel that's quite well written called Intuition by Allegra Goodman in 2006, where the main character is studying in the laboratory infecting tumors with viruses. Unfortunately, the novel is more centered around misconduct in science, but was an interesting read nonetheless. There was an episode of House in 2006 called House versus God, where they highlighted a herpes infection that caused a tumor shrinkage in one of the patients. Of course, everyone knows I Am Legend, the Will Smith movie, again, where they had cured cancer with a measles virus, but unfortunately things didn't turn out well for the human race in that. And then Where the Wild Things Are was an episode of Grey's Anatomy in 2008, and for several episodes during that whole season, they were conducting clinical trials in brain tumors by putting a virus into patients' brains. So Some of these are obviously raise the fear of using viruses, especially the I Am Legend movie, and hopefully by understanding some of the science behind this field, we can allay some of those fears. I don't want to minimize certainly the problem that viruses have caused to humanity over the ages. Smallpox and others have wreaked a lot of havoc, but I think we are getting to the point where there are enough alternatives out there that aren't as dangerous as some of those so-called hot viruses, that these can be used safely in people. So the scientific interest in this field has also been gaining attention. It's a perennial hot topic at the annual meeting of a variety of different organizations, actually, American Association for Cancer Research, but primarily the American Society of Gene and Cell Therapy. And then the Mayo Clinic has been sponsoring every 2-year boutique meeting with oncolytic viruses, and we have one coming up in March this year. And there was a virus approved, an oncolytic virus approved in China for use in head and neck squamous cell carcinoma in 2005. And I think it's only a matter of probably a few short years before the FDA approves a trial in adults, since there are several phase 3 trials ongoing now, assuming they have successful results. There have been a couple of scientific books written on the topic, and as you can see by my graph there, there's really a market increase in the recent 5 years, 5 to 10 years in publications in this area in the medical literature. So why the appeal of oncolytic viruses for cancer? For me, it's because they have multiple different modes or mechanisms of action. They can infect and kill the cell directly. So onco is for tumor and lysis is for cell killing. They can spread themselves, so self-propagation from one cell to the next to the next. They can induce an anti-tumor immune response. So they'll certainly induce an antiviral immune response in most cases, and if the infected cell is being killed by the immune system, then that's an if that infected cell is a tumor cell, then that will be killing the tumor cell. But it can also sensitize the immune system to tumor cells that aren't infected and therefore function as what we would call a cancer vaccine. And then most viruses are relatively easy to manipulate these days to engineer them to be increasingly selective and safe. But there are a lot of unknowns in this field, and I think one of the things I don't want the message to be in this webinar is that we know everything or that these are actual therapies yet or treatments. These are research at the moment, and we don't really know where their place in cancer therapy is going to be. I'm quite hopeful and optimistic that there will be a place for these at the table in terms of options for use in therapy, but you know, are they safe to use in people? Which ones are safe, and what doses? What are the short and long-term side effects? Will they improve survival or even cure patients? What are the best doses to use, dose schedules, routes of delivery, the settings? Should it be intratumoral, intravenous? Should it be in the setting of the operative resection of a tumor? What viruses would be best to treat or to use for which cancer types? Will there be certain ones that work best for certain types? Can we exploit their use as a cancer vaccine, maybe to prevent relapse by harnessing the immune system? And can the effects of them on tumors be improved by combining them with other therapies? As you know, most cancer therapies are used in combination, and no single approach is the answer to cancer. So in conclusion, from my introduction, I think there's a significant historical and theoretical promise of this field. There's certainly a growing interest among public and professionals. Numerous clinical trials in adults have been launched in the last decade, and I'm pleased to say that 2010 was sort of the beginning of the modern pediatric clinical trials, both through Children's Oncology Group, as you'll see, and through some efforts of others. And I think though we're at the very beginning of our learning curve, and there's a lot of basic research still needed to inform future clinical trials. Scott?
Great. Thanks, Tim, for the great, robust general overview of oncolytic viruses. And we look forward to hearing you come up again describing 2 specific clinical studies that you're a PI of. Next, I'm going to introduce Dr. Michael Burke, who is a principal investigator of the Seneca Valley virus. Dr. Burke is a pediatric oncologist, also an assistant professor in the Division of Pediatric Hematology Oncology, Blood and Marrow Transplant at the University of Minnesota. The Seneca Valley virus was one of the first, if not the first, oncolytic viruses to open in the clinic for pediatrics, and Dr. Burke was one of the first PIs to give a child an oncolytic virus in the U.S. So Dr. Burke?
Okay, great, thank you. We'll get right in here. So I'm going to essentially review some of the adult phase 1 Seneca Valley virus trial and then spend the rest of the time on the COG phase 1 that's currently open and enrolling. So Seneca Valley virus is considered a native, non-engineered, so non-manipulated virus, belongs to the Vaccinia family. And it's been developed as an oncolytic agent for human neuroendocrine cancers. These are cancers that are either neural in origin or may be hormone expressing. These cancers are particular targets for Seneca Valley virus. Precautions viruses are very small. They're an RNA genome. They are not a DNA virus. And some examples that you could relate to would be the rhinovirus and enteroviruses. And as was mentioned just previously, these viruses are essentially really non-pathologic in humans, the Seneca Valley. And those that are pathologic typically have mild illnesses, as in common cold and diarrheal illnesses. So NTX010, which is the name for Seneca Valley virus, was actually discovered really on accident as a contaminant that was identified while culturing cells. This was identified from porcine or pig trypsin, an enzyme used to break down tissue during the cell culture process, as well as in FBS, or fetal bovine serum, which is a culturing media to keep the cells healthy. Seneca Valley virus is given intravenously. It was infused at very high titers into mice, pigs, monkeys, and humans, and had no evidence of disease in any of these species and no dose-limiting toxicity in any of the previous studies. NTX010 was not transmitted between co-house adult mice studies. Essentially, they infected one mouse and housed it with another and checked antibody development in the uninfected mouse, which was not present, as well as sectioning the tissue of the uninfected mouse for evidence of virus, which was not present. Also, there's been no evidence of transmission between laboratory personnel using this virus or testing caregivers during the adult phase 1 trial. There's no transmission. So this schema here shows why viruses like NTX010 would be very desirable for oncolytic viral therapy. One is it's very small and has a good ability to penetrate a tumor mass. It replicates quite rapidly, and through that replication causes direct cell lysis. It's not a DNA virus. It does not get into the host genome. It is not pathogenic as well, so it does not cause disease in humans. It has a very low incidence of pre-existing immunity. In fact, I'm only aware of one person ever screened prior to receiving the virus that had an antibody. It was a farm worker, likely had exposure to pigs. So it's very low incidence of pre-existing immunity. A simple IV infusion. So this is, you'll hear about some direct tumor injections of virus. This is an IV administration, and this is a non-manipulated, non-engineered native virus, and it doesn't revert back to a pathologic form. It's very selective to those tumors that it targets, and quite effective in killing. It has a very benign safety profile, is what you'd like, and it's very easy to make, and FDA-approved cell line. So it sounds great. So the adult phase 1, there were 30 patients that were dosed in this study. Very different tumors typically than what we see in kids. Primarily they're small cell lung cancer and carcinoid, and then some other various neuroectodermal tumors. Patients were enrolled. They had been actively progressing by RECIST criteria, which is a general classification of response that's used for clinical trials. The dosing range covered 1 × 10⁷ to 10⁻11 viral particles per kilogram. On this study, there were no dose-limiting toxicities or drug-related severe adverse events. The drug-related adverse events were very mild, transient flu-like symptoms that lasted typically for 1 to 2 days. And there was not a clear relationship between viral load and side effects, meaning patients at the lower viral load may have had side effects or more side effects than those at the higher. It wasn't necessarily dose-dependent. The small cell lung cancer patients actually had a 3 times improvement in overall survival compared to historical controls that had equivalent third, fifth line similar treatments. Carcinoid patients had a 70% response. This was the largest response group by tumor type. There was partial response by PET, near partial response by CT, and some stable disease. So this picture is a CT scan of a patient with a carcinoid. The top picture shows 1 week prior to receiving viral infusion. The arrows point to 2 fairly large lymph nodes that were involved with carcinoid. And then 6 weeks after viral infusion, a single infusion, you show essentially resolution of the tumor. So there were essentially 10 evaluable patients, and these responses are all defined by RECIST for CT MRI. Minor responses by CT were seen in 3, stable disease, with a decrease in a tumor marker, chromogranin A, in 4. So some of these patients actually had tumors resected after viral infusion, which is not required on the pediatric trial through COG, but in the adult trial, some of these patients did have tumor resection, and so they had direct staining to see response. 2 patients with no response and 1 progressed. And there's a number of patients actually experienced prolonged stable disease, and some patients had improvement in symptoms, such as pain. This schema, the graph to the left, see if I can use this, there we go. This is blood, viral particles measured in blood or serum, showing that essentially all patients in this trial by 2 weeks, 14 days, had cleared virus. The next one is stool, which also was followed, and all patients essentially by 2 weeks, except for 1, had cleared stool, and all patients by the 1 month time of the study. This schema here is antibody development against the virus, and all patients developed antibody against the virus, which then further led to clearing through the stool and blood by 8 days or 1 week's time. Body fluid is essentially that this virus is secreted, is essentially the blood and stool. So NTX010 was detected in the serum and stool after dosing in all patients, and the viral kinetics were indicative of replication in the patients in the phase 1. It was cleared from all compartments between 2 and 4 weeks. All patients developed neutralizing antibodies by 1 week, and infectious virus was not detectable in serum after antibody development in these patients. So this is a nice proof of concept, a selective viral replication within the tumor. This, unfortunately, is autopsy of an adult patient who had small cell lung carcinoma, and you have 2 sections from the same liver, one that was involved with a metastatic site grossly, and one that appeared to be normal. And staining for virus, you can see to the left, normal liver, there's no, take my word for it, there's no virus that's being stained, and to the right, the brown little circles, that's all Seneca Valley virus. So now, overview of the COG phase 1, ADVL0911, Seneca Valley virus, which is currently open. Just a reminder, this trial is a recipient of the ERA Act Now funding. So there are 6 eligible diagnoses for patients: neuroblastoma, rhabdomyosarcoma, Wilms tumor, retinoblastoma, adrenocortical carcinoma, carcinoid tumor. Some of those may be obvious as neuroendocrine tumors. Others have certain proteins that they express that this virus targets, has been identified preclinically, and so is eligible for this study. Patients have to be 3 years or older and 21 years of age or less at time of enrollment. So Seneca Valley is given as a 1-hour infusion. It's a single dose on this study, followed by at least 28 days or until complete viral clearance from both compartments of serum and stool, whichever is longer. To date, there's been no patient that has not cleared virus by the 28 days. It's a single dosing on this study. Here's the very complex design. So the correlative studies, blood will be collected to monitor for developing of neutralizing antibodies. Archival tissue, so we do not require tissue to be resected at enrollment. So prior tissue that establishes a diagnosis of or at relapse will be submitted to evaluate for markers of neuroendocrine expression and potential expression of cell surface determinants of tropism, type 1 interferon signaling pathway members, and interferon stimulated genes, which you now can see how impactful they may be based on Dr. Kripe's nice introduction. Selected biomarker expression from tumor tissue will be correlated with tumor response, survival, time to progression. The concept of circulating tumor cells, occult circulating tumor cells, is actually no longer novel. It's quite well known that, particularly in metastatic diseases, that there are circulating tumor cells. In this study, we'll try to identify the presence and whether or not they're permissible to the Seneca Valley virus, meaning do they actually penetrate in and replicate and kill. So this will be assessed if present. This study opened at a dose level 1, 1 times 10 to the 9th. Enrolled 6 patients at that dose level. We're currently in dose level 2 at 1 times 10 to the 10th viral particles per kilogram. We will go up to 10 to the 12th and won't go beyond that based on adult studies have not gone beyond the 12th. Patient monitoring until viral clearance. Again, I had mentioned we will continue to follow these patients until they clear both compartments. It will start twice weekly, every 3 to 4 days during weeks 1 and 2, and then at least once a week, but not more than twice a week until viral clearance from each compartment. Toxicity profile is based on experience with SVV001 or NTX010 in adults, as well as our own trial, which we've learned something, which I'll mention, and general experience from other oncolytic viruses in clinical studies. So toxicities that we are expecting, fever and other flu-like symptoms. This is general oncolytic viral type of symptoms of chills, fatigue, nausea, vomiting, headache, diarrhea. Hypotension is something that's also been identified in other adult oncolytic virus therapies. Neutropenia is not something that had been seen on the adult phase 1 trial for Seneca Valley virus, but something we've noticed on our COG trial, so now this is an expected toxicity. So to end, we've enrolled 6 patients at dose level 1. You can see the majority of them have been neuroblastoma, 4 patients, 1 carcinoid tumor, 1 rhabdomyosarcoma. Dose level 1 was originally set for 3 patients. The first patient, a neuroblastoma patient, had a dose-limiting toxicity for pain. This was unexpected at the time. It was pain at the site of disease. It's now been, the study has been amended that this is not an unexpected toxicity. No further DLTs were identified in that first dose level. Moved on to dose level 2 at 1 times 10 to the 10th viral particles per kilogram. There's been a patient with neuroblastoma and adrenocortical carcinoma. And we are, again, have not hit another dose DLT and are waiting for the 3rd patient prior to moving on to dose level 3. And the study is ongoing. And that is my final slide, so we'll take questions after.
Thanks, Dr. Burke. And the questions should be submitted through the chat to the presenter section. We've gotten a few questions submitted. We appreciate that. So keep the questions coming, and we're gonna collect them and present as many as we can to the 4 presenters near the end. Those that we don't cover, we will have them follow up with us, and we'll present those back to you with an email of the questions and the answers that were not covered during the Q&A. I'm going to go back and reintroduce Tim Kripe again. He's going to be covering 2 sections because he's the PI of 2 different clinical studies. The first is HSV1716, which is herpes simplex oncolytic virus, and the second JX594, which is the Vaccinia oncolytic virus, both of which are at Cincinnati Children's Hospital and a couple other institutions as well. Lastly, I'll try to remind everybody that's presenting, we're going to try to fit 4 more presentations on these agents within the next 20 to 25 minutes. We have enough time for all the questions and answers. Without further ado, I'll give it right back to Dr. Kripe.
Thanks, Scott, and thanks, Mike, for the great presentation. Very exciting. I will have to say I've seen some of the questions go through, and want to remind people these are all early phase 1 trials. The purpose is mainly for safety, and we aren't going to be giving any information about efficacy. We don't really know that yet. It's pretty early. We're at the lowest doses and so forth. But obviously, with any kind of novel agent, the main concern first is making sure that it's safe. So the first study I'm going to talk about, and I'll try to go relatively quickly, is herpes simplex virus. As you know, herpes simplex causes canker sores, mouth sores, and can cause other diseases like encephalitis and other problems. The version that we're using is also called Seprehvir. If you took the vir, VIR, and blocked that out for virus, the rest of it is herpes backwards. So this is a mutant HSV 1716 that is provided to us from Crusade Laboratories in the UK. And this trial was launched with the help of Solving Kids Cancer. And with their generous support, we were able to get the trial started before we had any other funding. And it's now being collaborated by or funded by the FDA Office of Orphan Products Development. It's only open at Cincinnati Children's, being technically demanding and so forth. We felt it would be safer to at least start with a single institution study. We've enrolled 3 patients to date. Why HSV? There are a number of reasons. There had already been shown in adults that some of the deletion mutants, such as 1716, were clinically safe to put into adult brains and other locations. In the laboratory, they seem to be very highly lytic for cancer cells. They can wipe out a culture at a very low level of virus being put onto the cells within a few days. The whole culture can be wiped out. They're a large genome that can accommodate other genes. There's a defined clinical spectrum that we know about in people, and there's anti-herpetic drugs available if needed. And they do infect a broad range of cell types, even though most people think of them as mainly infecting nerves. The nerves are where they go latent, but they can infect pretty much any cell in the body if the conditions are right. And in some models, they've been able to induce an anti-cancer immunity and therefore may affect tumor sites that are not directly injected. This is a map of the genome. It's a 150,000 base pair double-stranded DNA genome, and it has various structures, which I won't detail, but the gene that's been deleted is present in 2 copies, and both copies have been deleted. That gene has been sometimes referred to as the neurovirulence gene, because when it's deleted, the virus no longer seems to cause encephalitis. It can still replicate in some normal cells at a low level. There have been some recent reports of that even in some brain cells. But it's been shown to be safe in adult trials to be injected at the doses tested so far into patients with brain tumors. So in mice, a normal wild-type herpes virus that's just found out in nature is lethal, down to as few as 10 infectious units or particles when put into the brain, but more than 1 million have been safely put in to the brain of mice without effects. And intravenously, we did a 2-year toxicology study in mice. I noticed one of the questions was what kind of data were required by the FDA to get this approved that was pediatric specific, and mostly they just wanted toxicology studies by the route that we were planning to use. So we needed to do some deep tissue injections, intraperitoneal, and also some intravenous injections in mice. And we used a dose that was predicted to be 100-fold above on a per kilo basis what we would use in the patients as proposed in the trial. So they really only required safety data by the route that we intended to use in the patients, not any specific pediatric efficacy data in cell lines or tumor models. Nevertheless, we have data about that numerous cancer cell lines are killed by this virus, including a number of pediatric lines, and there's anti-tumor effects in a variety of different mouse models where if you inject the virus, you can get tumor shrinkage and in some cases cures. So the clinical data in adults, there were over 70 patients treated in Europe on several different clinical trials, mostly patients with brain tumors, but also some patients with melanoma and a head and neck squamous cell carcinoma. Been administered up to 2 million infectious units at a shot in both single and repeated administrations with only minor toxicities experienced. And in some cases, scientists were able to resect or remove some of the tumor that had been infected and measure that the virus was actually replicating in that tumor. And most of these early phase studies aren't going to look very detailed at how well it worked in terms of fighting off the tumor because they're not designed to do that. We start at the low doses, and patients are way advanced in the stage of their disease. But many patients retrospectively live longer in some of those studies than were expected to live, for what that's worth. It's obviously worth a great deal. I don't mean to belittle it, but from a scientific standpoint, it's hard to determine whether that's a robust data set in terms of whether it really was working or not. So in this trial, we wanted to bring this technology to pediatric patients. And in this particular study, we were starting off with adolescents and young adults. The treatment plan is a single dose, but we felt it would be unethical to withhold future doses if a patient had a response. So we designed a part 2 with up to 3 additional doses if the first dose seemed to have an effect on shrinking a tumor. These injections would be at least 28 days apart, and there was an optional biopsy to look at whether virus is present in the tumor on those subsequent injections, since we'd have a needle there in the tumor anyway. The dose levels at the moment, it's only 2 dose levels. We've finished the first, and we're now recruiting for the second dose level at 2 million particles or infectious units to be injected. We expected, like Mike said, in terms of the Seneca Valley virus, some flu-like symptoms. We don't expect, just because it hasn't been seen in the adult studies, local or systemic herpes infection or encephalitis. We take some precautions, requires an overnight admission for monitoring. They're on contact isolation when admitted, and then at the next visit in clinic for follow-up. But after that, that contact isolation is removed, and standard precautions are used at subsequent visits. We recommend good hand washing and good hygiene for families and friends when at home. Again, we're only including patients aged 13 to 30 for this initial study. I hope someday that we'll be able to lower that age if we show that it's safe in this age group. We only require the primary cancer to be non-brain tumor at this point. I also hope to open a study of patients with brain tumors someday. We require a certain minimal size of the tumor in order to be able to access it with a needle under interventional radiology guidance by CT or ultrasound. So in this particular trial, the level that's been required is 1.8 centimeters in each of 3 dimensions, and it must be thought to be safe to be injectable by our radiologists. There must not be other infections or severe systemic diseases present. Patients cannot be on antiherpetic drugs, or these need to be discontinued for the month following the injection. And there are other standard requirements for phase 1 studies. So the virus is administered directly into the tumor. It's delivered as a slow push as we move the needle around, or at least pull it back over the needle track. And obviously we wouldn't do it unless we thought it were safe to do. So this is a table that perhaps you can refer to later that tells what the follow-up schedule is required, basically weekly visits for a month and then monthly for 3 months, and every 4 months thereafter. A variety of labs, and we're also looking at whether patients become immune to HSV, and it's not required that they be immune to it prior to the study, and as you know, most adults will be immune to HSV1. We also look for virus cultures and PCR assay for their genomes. So if anyone's interested in finding more, there's information on clinicaltrials.gov. They can also contact me or the lead regulatory coordinator in our office, or email there at cancer@cchmc.org. So Scott, if you don't mind, I'll just go straight into the next study. JX594 is a Vaccinia virus being sponsored both by Solving Kids Cancer and by Generex Therapeutics in San Francisco. This again is a phase 1 study similar to the one I just described with an intratumoral injection of this virus. The virus is again called JX594. It is now open both at Cincinnati Children's and Texas Children's in Houston. The first patient was injected. As far as I know, there's been 1 patient to date. So it's again quite early on in in the study. So JX594 is not smallpox. Patients and providers cannot acquire or get smallpox infection from JX594. So, but it is a gene-deleted version of cowpox, so it's related to smallpox, but it's derived from Vaccinia virus, the Wyeth strain that was used in the smallpox vaccine campaign that eradicated smallpox worldwide. So there's a large safety database of the wild-type Vaccinia virus, which was given to hundreds of millions of people, and so we know a lot about what it does. The version we're using, the JX594, has been genetically modified. It's been attenuated by deleting the thymidine kinase gene, which is thought to target it more to replicating cells. There's abundant thymidine kinase in cancer cells, and that enables efficient replication in those cells. That enzyme is not activated to much degree in normal differentiated quiescent cells, and therefore doesn't grow as very well in those cells. It does express, as I mentioned earlier, a transgene, a gene that expresses the granulocyte macrophage colony stimulating factor, GM-CSF, which is designed to help the whole infection induce an immune response against tumor antigens. This is a diagram of the genome. It's a little bit bigger than the herpes virus, 190,000 base pairs, double-stranded DNA genome. The thymidine kinase has been split and a deletion, and in its place is the GM-CSF gene, as well as a beta-galactosidase bacterial gene that is used as a marker, and one can look for the virus using that gene in tissues. There have been 6 trials that I'm aware of in adults, more than 90 patients to date, phase 1 trials in melanoma, hepatoma, or liver cancer, and now an intravenous trial in patients with a variety of different types of solid tumors. There's been a phase 2 study in hepatoma, and there's now an open phase 2 study using intravenous virus followed by intratumoral virus and combining it with another drug. So it's certainly advancing through the clinical trials, and certainly time to be bringing that technology to children with cancer. Some proprietary novel injection technique is being used in conjunction with Generex. The company developed this quadrifuse needle. As you can see from the picture here, it's the needle is able to be deployed once it's in the tumor to 3 different sites, each of which has 4 exit sites for the tumor, so it can help it spread during the injection at the time of delivery. Again, fever, chills, some pain, and other systemic nausea, fatigue, that kind of thing have been observed in patients, but it's very transient and usually is resolved within 1 to 2 days. This is an example of a patient. This example shows a couple of things. One, that this is a metastasis that developed in a liver cancer patient that was directly injected and shrank with a near complete response. And this patient had previously been treated in the liver with the virus and had high neutralizing antibody titers. So not only does this show that the virus can induce a tumor response, but it can do so in the face of a pre-existing immunity to the virus. So in our pediatric study, it's very basic. We're just trying to determine a maximum tolerated dose, that's MTD, or maximum feasible dose, that's MFD. That feasibility may be limited by how much virus we're able to give, and determine the safety and toxicity in this group of patients. Secondarily, we're looking at pharmacokinetics and pharmacodynamics. That's PK/PD. That looks at the levels of the virus in the blood and in any tissue, whether there's a response, and then to look at the immune response. So it's a standard design, a dose escalation. We're starting at 10 to the 6th plaque-forming units per kilogram and going up to 3 times 10 to the 7th. You'll see different numbers with different trials for these, and they're all unique to their own vector, their own virus. We do some follow-up scans at 3 weeks, and patients can get up to 3 more monthly injections at the same dose if they have a response. So again, this one now is open down to age 2, so age 2 to 21. Patients with advanced or metastatic non-brain tumor, solid tumors, they can have had brain metastases if they've been treated, but they might, if they have other options for cure, they shouldn't come on this study. The lesions can be done down to as small as 1 centimeter, and patients need to have no evidence of an significant immunodeficiency, such as high-dose steroids, immunosuppressants, diagnosis of HIV or AIDS, or known genetic immunodeficiency, and we screen patients with a CD4 count as a measure of deficiency. So the enrollment criteria should actually say greater than 200. That's an exclusion criteria, less than 200, meaning patients must have greater than 200. There should be no other significant conditions or antiviral or antiplatelet or coagulation therapy. So just to touch on the toxicity a little bit, and then we'll wrap up. Hundreds of millions of patients have been inoculated safely with the wild-type version. This is thought to be a version that's even more safe. It's been well tolerated, but the wild-type version did have some rare but serious complications in very rare patients. Approximately 0.005% of patients that were inoculated had some problem with things that are listed there, and disseminated rash, encephalitis, eczema, or myocarditis. And 1 to 2 million did die of complications from the Vaccinia infection. None of these have been observed after use of JX594 in the adult patients to date, but it does enable us to identify the highest risk individuals. So these individuals are not permitted to enter the trial at this stage, nor are they permitted to take care of patients within the first 3 weeks of being on the trial. So women who are pregnant or nursing, children less than 1 year old, people with severe skin diseases, because the virus can spread from a weeping skin disease, or those who are immunocompromised. Those patients are not eligible for the study. They should also not be in the same household for 3 weeks of patients who are enrolled in the study, and they should not be handling the virus or cleaning the rooms after the patients left. I provided this slide for you just to compare the two trials that we have ongoing here. Both of them are intratumoral, and they have some differences in terms of what's required for infection control. And again, the same contact information for us if you want to know more about it. Scott?
Thanks, Tim. And we will have the archive of this presentation available both on the website and through the email that we send up as a follow-up. So thanks again, Tim, for covering a lot of basis in a short amount of time. I'm going to move on to our next presenter, who is Dr. Corey Raffel. He's the only surgeon among the group. Corey's the chief of neurosurgery at Nationwide Children's Hospital in Columbus, Ohio. In addition to neurosurgery, his research interests include tumor suppressor genes, oncogenes in medulloblastoma, tumor stem cells in medulloblastoma, and now oncolytic viral therapy for pediatric brain tumors. Dr. Raffel is going to describe the modified measles virus for medulloblastoma. Dr. Raffel?
Scott, thank you very much. I'm really glad to be able to present this information to the group, and I'm going to have difficulty following the prior really great talks. I'd like to point out before I start talking that the phase 1 trial that we're discussing here has not even started yet. It's at the FDA. We don't have any patients enrolled. So what I thought that I would do was present the background data and rationale for using measles virus as a treatment for recurrent medulloblastoma. Briefly, I thought I'd mention medulloblastoma, which is the most common malignant brain tumor in children. There are about 350 new cases of medulloblastoma in the United States each year. At this time, we're having about 70% of the patients survive for 5 years after disease, although patients do continue to die after 5 years. The problem is that our treatment for this disease is really toxic. We have to use radiation therapy, and usually radiating the brain and spinal cord of these children, and this damages the developing brain, and the patients may suffer a decrement in intelligence. So it's a problem that we have reasonably effective therapy, but the patients that survive don't do so great. So we've been interested in oncolytic viral therapy and have been using measles virus. Just briefly to describe measles virus, it's a negative strand RNA virus with a very simple genome. It's a small virus. The two important proteins in the virus are the H-glycoprotein, which is the protein on the surface of the virus that recognizes a receptor on the cells. And there are two well-described receptors listed here, SLAM and CD46. And the virus makes another protein called the F protein, which will fuse an infected cell with another cell that's touching it, and that fusion results in the cells dying. The SLAM receptor is found on lymphocytes, but the CD46 receptor is frequently overexpressed on tumor cells, which gives us the opportunity to infect the cells with the measles virus. The strain of measles virus that we've been using is the Edmundston strain. This is a vaccine strain of measles virus, and unlike the normal measles virus, it has a real strong preference for CD46 over SLAM, and the Edmundston strain is able to replicate in human cells. I should comment that the Edmundston strain of virus has all the same advantages that Dr. Kripe mentioned for the Vaccinia virus. It's a vaccine strain, and millions of doses of this virus have been given to human beings with almost undetectable toxicity. So what I'm going to do in the rest of this talk is to try to discuss with you some of the work that we've done in our laboratory that's led to this phase 1 proposal. I'm going to try to show you that we can use the measles virus to treat medulloblastoma, that we can kill medulloblastoma cell lines, and we can also kill medulloblastoma cells when they're placed in the brain. So we did do some studies looking at some medulloblastoma cell lines. We looked at 5 cell lines, and all of them expressed the receptor. In addition, 12 of 13 samples that we removed from patients also expressed the receptor. So it looks like medulloblastoma would be a good target for measles virus, as the measles virus will recognize the cells because they have the receptor. When we treat cells in the lab with the measles virus at different doses, we're able to see whether the cells survive and whether they fuse together. And then we look to see whether the infected cells are making new virus. And this chart here is one cell line, and you can see that at a multiplicity of infection of 0.11 or 10, we're really able to kill the virus very effectively, whereas the untreated cells don't die. And here's another cell line that's even a little bit more sensitive to the measles virus. And this slide shows that while the cells are dying from infection of the virus, they're also producing new viral particles, and you can see that the production of new virus peaks between 2 and 4 days after infection. So once we were satisfied that the measles virus could kill medulloblastoma cells in the Petri dish, we wanted to see if it could kill the cells when they're placed in an animal. So what we did is we injected 1 million medulloblastoma cells from one of these cell lines into the caudate nucleus, well, part of the brain, in nude mice, which are immunosuppressed mice, or irradiated skid mice, which are also immunosuppressed mice, which allow for the growth of the human tumor in their brains. We let the tumor grow for a while. We put the cells in, and then 10 days later, when the tumor was established, we gave 6 injections of the measles virus into the same tumor site every other day, and then we measured survival. And you can see from this survival curve that when the tumors are allowed to grow either with no treatment or with virus that had been irradiated with ultraviolet light to inactivate it, the animals die pretty quickly. But when we treated them with the measles virus that is alive and able to kill the cells, we got dramatically prolonged survival. And here's a picture of what those animals look like when they're autopsied. The picture here is an untreated animal, and you can see where the tumor was growing on the right side of the brain, and here's a blow-up of that showing the interface of the normal brain with the tumor. And here's the brain of a mouse that was treated with the measles virus, and you can see it's hard to see any tumor. If we look really closely at higher power, we can see the needle track where the tumor and virus was injected into the brain, but we don't see any tumor at all. Another way that we track these tumors in the animals is we used cells that have a protein in them that, when treated appropriately, will glow and emit light, and we can take pictures of the animals and watch how much light is coming out of their tumors. So here is an untreated animal right after injection of the tumor, and you can see that they get progressively more red because more and more tumors growing, more and more light's being emitted as the tumor grows in the animal's head. And here's an animal that we start with the same tumor, but when we treat with measles virus, the tumor completely goes away. And here's an animal that's treated pretty effectively, but at the end still has a small amount of tumor. And interestingly, this tumor was actually at the injection site, so a few cells had leaked out and weren't where we were injecting the virus. So we conclude from this that medulloblastoma cell lines express the measles virus receptor, and that they're killed effectively in the Petri dish by the virus, and that when the cells are killed, we get virus replication, which means we get more virus to kill more cells. We also demonstrated that samples from patients express the virus receptor. And we also showed in an in vivo model that if we gave the animal a tumor, we could treat it effectively and cure the animals of the tumor. So that's led to our phase 1 trial. Now this is a phase 1 trial. It's a toxicity trial, and the proposal is right now working its way through various regulatory bodies, both here at Children's Hospital, at Nationwide Children's Hospital in Columbus, at Ohio State University, which is our parent organization, and also through the FDA. Our plan is to treat patients with recurrent medulloblastoma by resecting the recurrence and then injecting the virus into any remaining tumor or the tumor bed. I'm hoping that we'll be able to have this trial open within a year, although bureaucracy being what it is, one never knows. And I just comment in closing that we've also been able to demonstrate effective treatment of medulloblastoma in a murine model of CSF dissemination. That's when the tumor cells float off into the fluid around the brain and spread along the brain and spinal cord. And we are in the process of writing that phase 1 trial also. Thanks for your time and attention.
Thanks, Dr. Raffel. We'll be crossing our fingers that the trial can open sooner than the bureaucracy would delay it. So
yeah, thanks, Scott.
Hopefully in the next few months. Our final presenter is Dr. Andy Kolb. Andy is a pediatric oncologist and also a director of the blood and bone marrow transplantation and head of cancer therapeutic laboratory at Alfred I. DuPont Hospital for Children in Delaware. And he is the principal investigator of the newest oncolytic virus that's going to be in the clinic sometime, hopefully this year, is the reovirus rheolysine agent. Andy, would you like to wrap it up for us?
Yeah, thanks, Scott. So I'm going to talk about the phase 1 study of reovirus in patients with relapsed or refractory solid tumors. This is a NCI and CTEP and COG sponsored trial through the COG phase 1 consortium. So it's open currently at a variety of different sites. It opened at the end of December. We have yet to enroll our first patient. Most sites are still contending with the IRB approval, but it should be open at many sites within the next few weeks to months. You can find sites where the trial is open through the clinicaltrials.gov website, and the trial identifier is listed there. And I put this slide in just to remind everybody that this, along with all the viruses that you've heard about, are manufactured products regulated by under general manufacturing practices and through the FDA, so that these can be administered safely as a drug. Even though these are viruses, they are treated as a drug with added safety precautions to protect patients and staff. So rheolysine is an unmodified reovirus. This is a native virus, and it's a double-stranded RNA virus. What's unique about reovirus is that it selectively kills cells with activated signaling through the RAS pathway, and I'll come back to this in a minute. But the RAS pathway is utilized by a variety of different oncogenes in many different cancer subtypes. Reovirus has been isolated from the human respiratory and gastrointestinal tracts. It's called REO, R-E-O, because it's a respiratory enteric orphan virus. And it's called an orphan virus because it's not typically associated with any symptoms from exposure. Back in the 40s and 50s, large doses of reovirus were given to prison inmates and not shown to be associated with any symptomatology in these recipients. About anywhere between 70 to 100% of adults show evidence of some prior infection in the form of antibody titers to reovirus. I'd mentioned RAS activation. I'm sorry, I think I skipped a slide. There we go. That this is production of infected viral particles is restricted to RAS cells, but this virus can infect any cell within the body. And it has a capsid which is dissolved upon infecting the cells, and then the core. And within that core of the virus are the replication machinery to allow the virus to divide within the cells. And that machinery is deactivated in cells that don't have RAS activation. Again, this is like the Seneca Valley virus. This is an RNA virus that divides within the cytoplasm. No gene is inserted within the human genome of cells that it infects. RAS can be activated in cells through a variety of different mechanisms. There are receptor tyrosine kinases like EGFR, IGF-1R, PDGFR, which can be dysregulated. They can be dysregulated by mutants in the receptor tyrosine kinases, by overexpression of the receptor tyrosine kinases, or by overexpression of the molecules that activate these receptors. It can be activated within the cells, mutations that signal through other tyrosine kinases, or it can be activated through mutations in RAS itself. Mutations in RAS are uncommon in pediatric malignancies, but are seen more frequently in adult malignancies. They are not required for viral activation. This is a slide of a patient in a phase 2 trial that received reovirus. This was a systemically administered reovirus. This is pre-treatment. Here you can see this large lymph node in the neck and on CT scan. And then after 3 cycles, this patient had a near complete response. This patient was heavily pre-treated with standard therapy for head and neck malignancies. Sorry, that slide was a little out of order, but getting back to the RAS story, this is just a cartoon depicting the fact that in RAS-activated cells, the mechanism that allows viral replication is allowed to proceed. In cells that have wild-type activation through RAS or wild-type signaling through RAS, that mechanism is turned off. So viral replication proceeds preferentially in RAS-activated cells, lysing those cells, but allows normal growth in, does not allow viral replication in normal cells. This is a series of experiments that we did just in a couple xenograft models. So this is a rhabdomyosarcoma, a Ewing sarcoma, and 2 osteosarcoma tumor lines that are injected into mice. And we administered 3 doses of the virus during the first week. And you can see that in the control tumors and the control mice, so the black lines, the tumor growth is allowed to proceed. In the reovirus-treated tumors, there is some response, stable disease in 3 of the tumor lines and some progression in the 4th tumor line. And this is an electron micrograph showing viral particles within the tumor cells that were harvested 24 hours after systemic administration of the virus. So far in adults, there have been 16 completed or ongoing phase 1 and phase 2 trials of reolysin in the United States, United Kingdom, and in Canada. Patients, more than 290 patients have been treated to date with multiple intratumoral doses or IV doses, alone and in combination with radiation and chemotherapy. So the experience to date with reovirus is quite substantial. Almost 300 patients have been treated. What's interesting in these trials is, one, there have been partial responses and stable diseases, both alone and in combination with chemotherapy. There were some outstanding responses in patients with head and neck malignancies. So 19 patients with head and neck malignancies treated in a phase 1 trial and a phase 2 trial. 8 of these patients had a partial response, and 6 patients had stable disease, some upwards of a year on trial. So that's 14 out of 19 patients with refractory head and neck malignancies that had significant responses to the virus. Among the 44 patients with recurrent sarcomas, there have been 6 patients with stable disease of more than 6 months duration. I believe 1 patient is actually still on study more than a year after starting. Also of note in these trials, there has been no maximally tolerated dose. So there are no dose-limiting toxicities that have been associated with this virus. And this is injecting up to 3 times 10 to the 10th viral particles into adult patients. This dose of 3 times 10 to the 10th was defined based on manufacturing limitations. So the company at the time was not able to manufacture a dose higher than this to get administered to the patients. There have been no adverse events other than flu-like symptoms, some mild and transient abnormalities in liver function testing, vomiting, diarrhea, and nausea. And as a result of all these trials, there's a phase 3 trial now in adults underway in patients with head and neck cancer of reovirus used in combination with conventional chemotherapy. This is another example of a response patient with a another example of a response, a patient with a hepatic lesion that shrank. So our trial design for the phase 1 trial is this. There are going to be 3 dose levels. The first dose level is about 60% of the adult dose adjusted per kilo. So the adult dose is a flat dose of 3 times 10 to the 10th. We're going to use 3 times 10 to the 8th and adjust that per kilo. And this is 60% of the adult dose, which was a safe dose. And then escalate to the full adult dose adjusted per kilo. And then the third dose level will be reovirus in combination with oral cyclophosphamide. And the advantage to this is that the dose of oral cyclophosphamide that we're administering is thought to suppress the immune response. So we're going to be able to evaluate in parallel cohorts whether or not we're able to prolong viral titer in the patients, prolong viral titer and inhibit the antibody response in patients. The eligibility requirements include aged 3 to 21 years of age, relapsed refractory solid tumors with the exception of lymphoma and CNS tumors. The reason for these exceptions is that we're not sure in CNS tumor patients how much inflammation will be associated with the administration of the virus, but this is definitely a priority for future testing. And patients can have received no immunosuppressive therapy like steroids for 6 weeks prior to enrollment on the trial. We're going to be administering the virus via intravenous administration daily for 5 days and repeat these cycles every 28 days. It's a 1-hour infusion. We're going to be looking at viral titers in by PCR in the serum, in saliva, and stool, and we're also going to be looking at reovirus titers. And this is a phase 1 trial, so we're going to be looking for dose-limiting toxicity and recommended phase 2 dosing and schedule. And as I said, the trial is open for enrollment, and you can see what sites are open at the clinicaltrials.gov website. So thank you.
Thanks a lot, Dr. Kolb. I'm going to jump right into the Q&A so we can try to fit in as many of the great questions we have as possible. And again, those that aren't covered, we'll be archiving all the questions and answers and sending them out in an email that will also include a survey for your feedback and the link to the replay archive of this entire webinar, both audio and slides. The first question I'm going to jump into probably for Dr. Burke and Dr. Kripe is about safety and toxicity in general. It would be, how do I know if a particular trial is safe for my child? And secondly, can you compare toxicities between these viruses?
Mike, you want to start?
He had to leave.
Oh, okay. So this is Dr. Kripe. I guess we don't know whether something is safe for any given child or not. The trials have been carefully vetted through multiple different groups, local IRBs, in some cases national committees, certainly companies that are involved with their own clinical trial specialists and medical leadership. And so the reason that you see many of the restrictions are designed to exclude patients where we think there's a high likelihood of a problem. So many of these initial trials have excluded patients with brain tumors. The worry is that there might be some transient swelling and inflammation associated with virus infection that could cause problems within the brain. Now, Dr. Raphael's strategy is to embed it into the tumor bed after resection, so that seems very reasonable, a way to minimize that risk. And so the purpose of doing phase 1 studies is really to determine if these are safe. We are confident or cautiously optimistic that these viruses will be safe based on adult experience, based on studies in animal models looking for toxicity. But that is why we're doing these phase 1 trials, to really determine that in people, in real live people, whether they're safe or not. And therefore, this is very early phase research. And if they're shown to be safe, or if we define certain doses that we need to stop at in order to keep them safe, then we will be able to go to more extensive trials to look to see if they really work.
Just a follow-up to that from one of the participants is, do genetically modified viruses cause more potential risk because they're genetically modified?
Most of the viruses that are genetically modified are modified in a way to decrease their infectious capability. So they're mostly mutations of genes that are critical in normal cells. And so, in fact, they're thought to be more safe than the wild type or normal, naturally occurring versions.
Okay. Next is about administration. I'm not sure if everyone understands that some of these trials have intratumoral, which is directly injected into the tumor, while some of the other ones have an IV or systemic administration. Can you quickly explain the differences between the two and if there's any advantages or disadvantages? I'll go back to you, Tim.
Sure. The reason we started with intratumoral is we thought it would be the safest route to start with. And certainly with the JX594, the adults have now gone into intravenous, and we certainly hope to be able to do that as we move forward. And in fact, with that virus, and I understand with the Seneca Valley virus, both of them tend to be spread through the blood anyway after intratumoral injection. So in effect, one's creating a virus factory by doing an intratumoral injection that sort of feeds the blood with the virus. So in some ways, it may be similar to intravenous. Certainly intravenous is more likely to reach other distant sites of disease, metastases, and that's going to be very important for most patients. And so at the moment, the reason there's a couple that were intratumoral, it was thought to be the most likely to have an effect and the most likely to be safe. But certainly there's hope that in the future those will be able to be used intravenously. I think the two that are being used intravenously now are able to do that because they've already shown that to be safe, a safe route in adult patients.
Yeah, I think just to add to that, Tim, for reovirus, the trials have been ongoing for about 5 years now, and all the initial intratumoral administration trials showed that the virus was indeed safe, and that allowed advancement to intravenous administration. The big disadvantage of intravenous administration is that you have to wait for the virus to circulate, and for these viruses where there is a potential for an immune response, you may decrease some of the drug delivery that way. You're injecting a huge quantity of virus, which is thought to maybe overwhelm any antibody-mediated response, but nonetheless, it certainly affects the delivery of the virus to the tumor.
Dr. Kolb, I'm going to go back to you with another question. This is about efficacy in general. How does a virus treat metastases if it's injected locally into a tumor?
In some of the trials with reovirus, for example, there was evidence of distant responses with intratumoral administration. So there were a couple patients with head and neck malignancies that had regression of tumors, either pulmonary or lymph node mets in a different site. These reports are anecdotal. There are only a couple of them. But the idea is that the tumor itself is a factory to continue making virus, and that there may be some systemic spread of the virus from that site. There may also be some direct, unintentional, but direct injection of the virus into the venous system at the time you put it in the tumor. And both those methods may allow the virus to spread systemically, but it's certainly not the intent with the intratumoral administration.
And this one would be for either Dr. Kripe or Dr. Cole, but can my child enroll in a second oncolytic virus trial if enrolled in a prior oncolytic virus study?
Right now, at least for the COG trials, there is a limitation that the patient should not have received another immune modulating or viral therapy.
So with the HSV and the JX, there has to be a 28-day washout period from previous, but patients can get one virus and then another.
Okay. Dr. Raffel, this is a question about other CNS tumors. Is there anyone planning on a trial that will include other CNS tumors besides medulloblastoma that you're aware of?
Well, the measles virus has been trialed in adults with glioblastoma multiforme recurrences, and I think that we do have the possibility of opening the trial up for tumors in the astrocyte line of tumors in children. We haven't done the in vivo work to demonstrate efficacy on pediatric tumors. We're especially interested in doing brainstem gliomas, and some lab work has started in that regard.
I'm not sure who to direct this question to, but I'm just going to throw it out. Is there a risk, are there any risks of treatment with viruses if the tumor is involved with an important blood vessel or involved with the heart or other sensitive areas of the body? Is there a risk of bleeding out if the virus is very effective or swelling?
I think those risks exist for any therapy that you would administer. There may be some added risk of swelling at the site, and this is for the viruses that are administered systemically. There may be some risk of swelling at the site. I certainly think for intratumoral administration, you have to take the site of the tumor into account prior to enrolling on the trials.
Yeah, and in fact, we've had some patients we weren't able to enroll on the trial because the location was felt to be too risky.
Okay. So you would assess each tumor location as being safe or not safe to inject, no
Correct.
matter where it is, right?
Right.
Okay. Let me ask this other question. So if there's several trials, to choose from, it looks like there's going to be 5 in 2011 and maybe some others that we don't know quite yet. How would a parent approach choosing which trial is the best for their child? I'll ask Tim first since he's a PI of 2 different trials.
So in some regards, it's self-selecting because each trial has their own specific criteria, and they're not all the same. And one reason they're not all the same is because they've basically grown up differently. They've been either written by different people or different groups of people, and they have different inherent issues that have been raised by adult studies, and so the criteria are not all the same, and so a given patient may not qualify for all of them, and if a patient does qualify for 1 or 2 of them, I think the idea would be which might have more data available for that tumor type, either in preclinical studies or other or if it's later in the trial, perhaps there are other patients. So it's going to be an individualized decision between the treating oncologist and the investigators on any one of these trials, and there may not be, all of them may not be an option. Again, for example, the Seneca Valley virus is for neuroendocrine tumors and so forth. So each of them have their own criteria, which is why I think it's very helpful that you have that table there on that board. And as we move forward and open more and more trials, hopefully these criteria can be loosened and we can enroll more patients.
Okay, great. Just a couple more questions. This looks like it's for Dr. Kripe. Does the herpes or vaccinia virus integrate into the host genome?
So neither virus does. So there's not a risk of a long-term mutation by integrating into the genome. The vaccinia, although both of those viruses are DNA viruses, and most DNA viruses do replicate in the nucleus where the cellular DNA is, the Vaccinia virus is unique in that it acts more like an RNA virus. It replicates in the cytoplasm, so there's virtually zero risk of it incorporated into the cellular DNA and permanently changing that cell. Herpes virus does go latent in cells, particularly in neurons, and the virus persists for long periods of time in a quiet state. But it exists as an episome, meaning it exists separately, like its own chromosome, so it doesn't integrate into the genome. So this is a great question in terms of safety, because retroviruses that are used for a lot of gene modification trials do integrate into the genome, and that's what they're designed to do, to permanently change the gene to cure a genetic disease. And sometimes patients have developed problems with that, but that is not really a risk for these viruses. And in fact, all of them are lytic viruses, so if there's an infection, they're likely to kill the cell anyway and won't be an issue.
Okay. Dr. Cole, I've got one related to the reovirus. It's which sarcoma had the lower response rate of the sarcomas you had put up on the presentation?
That trial, those trial results were presented at ASCO in June, and there were still a couple patients who were on the trial at the time. Majority of those sarcomas were adult patients. So this phase 1 trial, I think that trial went down to age 13, but the youngest patient they enrolled was 18 or 20. So we're talking probably a different group of sarcomas than what you would see in pediatrics. There were no patients with rhabdo or Ewing sarcoma that I can remember. There were a couple patients with osteosarcoma. But the responses that they saw were in patients that had other adult-specific sarcomas like synovial sarcomas, which at least are more common in adult patients. And there weren't enough responses to really say which sarcoma is most likely to respond. We've seen responses in the lab in a variety of different tumor types.
Okay, great. I'll ask either any of you, because you're all working on phase 1 trials. The general question came up about what would the phase 2 study trial design look like for any of these viruses?
I think for the reovirus trial, it's already there have already been some phase 2 and then now the phase 3 as well. And I think the interesting question that I saw somebody had mentioned in the chat window is what happens when you combine these trials with chemotherapy? What happens when you combine these trials with immunosuppression? The idea being that you would inhibit the body's innate ability to clear the viruses. You may also inhibit the interferon response, and there's some advantage to that, as Tim had pointed out initially. So the phase 2 trials, I think, for at least for the Seneca Valley virus and for the reovirus, may be centered around prolonging viral clearance from the body, so that using the drugs in combination with conventional chemotherapy, inhibiting antibody response, may allow the virus to hang around a little bit longer.
Okay, just a couple more questions. We're doing pretty well on time, just about 15 minutes over. This is for any of the researchers. Do you know if there's any bystander effects that have been seen so far or anything that could be predicted?
So bystander effect traditionally has meant effects on cells that are not the infected cells, so cells surrounding it, and that became a popular term when there was the concept of using this prodrug enzyme, a thymidine kinase gene, actually from herpes virus, and then administering the drug acyclovir or ganciclovir to kill the cell. And it was found that the activated drug was then able to migrate through connexin or connective channels between cells and kill cells around it. So I broadened the definition, though, in my mind, of being able to affect any cell that's not infected. So the immune response that's elicited by a virus infection to tumor antigens that are present on all the tumor cells, not just not just infected cells, would be considered a bystander effect in my book then, so that cells that are not injected might be affected. So I think there's the probability that all of these viruses will induce not only an antiviral immune response but sensitize the immune system to tumor antigens. in the context of an inflammatory microenvironment with the infection. When the cells die, these different proteins in the cell are exposed in the right kind of environment to be, induce an immune response. So in that sense, I think there's a high potential for a bystander effect. But in the traditional sense of a cell being affected that's not an infected cell, I don't see how that would happen.
Okay, and I'm going to wrap it up with one sort of practical question that families might find helpful about logistics. And these trials tend to require patients and families to stay over potentially for a period of time, and also some traveling from out of the area because they're not available at all different institutions throughout the U.S., sometimes just a small number. And if you don't have the information on this, that's great. We can follow up and get the information back from other areas of the hospital that may have it. But it's about housing and taking precautions for staying over for oncolytic virus treatment. Do all of you have Ronald McDonald houses or any other facilities available for families that would be traveling from outside and potentially staying longer than 1 to 2 days for treatment or monitoring? I'll start with Tim first.
Yeah, at Cincinnati we have a large Ronald McDonald house, and that would be okay for the herpes virus trial, but the Genomic trial, the Vaccinia trial, requires somewhat isolation and precautions. So we're not allowing patients to go to areas where there might be immunocompromised patients. So someone from out of town either needs to find a place to shack up, or to we have some arrangements with some of the local hotels, and we do have some funding available, and through our social services, some assistance is available to help with defer some of those costs.
Dr. Raffel?
Yeah, we also have a Ronald McDonald house, and everything that Dr. Kripe said applies here too.
Dr. Cole?
Yeah, we have a Ronald McDonald house. We didn't go into that. Mike talked about it briefly, but the shed and spread of some of these systemically administered viruses that have been through trials has been minimal. So a few patients have demonstrated viral genomes in their urine. It's not clear whether or not those were live viruses or lysed viruses, destroyed viruses. So we think it's relatively safe to staff members and family members around them, and patients are permitted to stay at our local Ronald McDonald house. I think a lot of the hospitals that participate in these multi-center trials have Ronald McDonald houses in the community, and it's relatively easy to find out through the internet.
Okay, great. Well, I'm going to wrap it up and say thank you very, very much first to our 4 presenters, Dr. Burke, who's no longer on the line, Dr. Kripe, Dr. Kolb, and Dr. Raffel. I'd like to also thank Donna Ludwinski from MB Globe, who was very helpful in putting this meeting together as a coordinator with all of the researchers and presenters and all the families that were able to attend. And I want to thank all the families as well as the researchers who took the time out of the middle of the day to spend an hour and a half with us here. It's really great because there's power in numbers for all of us to hear cutting-edge information about therapeutics that can potentially help our kids, and be able to ask great questions and have them all in the same room and be able to share these questions and answers with you as a group, which is part of what we're going to do when we follow up in the next couple of days with our email, which I've mentioned. We just ask that you complete the survey that we have, which should probably be between 5 and 10 questions, fairly easy. We'll include the archive link, which has the entire replay of the webinar, both audio and the computer, the slides, and then the questions and answers, which we'll have available as an archive as well, printed out. So thanks, everyone. I really appreciate you coming together to make this happen. It's a really exciting time in 2011 that we have these new options that are exciting and still early stage, but we're really hoping that these can make a big difference. We definitely know that they're less toxic, which is a great new option for kids. So thank you once again, and look forward to having another follow-up presentation on another subject probably down the road in 2011 again. So thanks, everyone, and have a great night.
Thanks, Scott.
Okay, yep, thanks. Good night.
So long.